Hey, welcome to Unknowns. This is something I want to do long back, and today I found some time. So here it goes. Many times in microbiology, I hear students blaming on the laminar or their environment, saying that their plates got contaminated. There is no such things actually. Either you lack a skill to make a clean control. Okay, that's the only truth it can exist. Okay, you should know how to handle the plate in order to produce a clean control or in order to have a sterile examination. Okay, basically microbiology lab is like, you need to earn the skill. What is the difference between writing a theory and conducting an experiment is this: you can read about producing clean control in thousands of books, but you can never produce it unless until you sit in the laminar, work with your plates, produce a lot of negative results, then you see where you are doing wrong and then you are correcting yourself. Okay, there's no other means to produce a clean control. Just by reading a book you cannot do it. That's why I used to say theory and practicals in life science is completely two different game. Okay, you may excel in theory without knowing nothing about your practical. Okay, so until unless you spend your time and energy to earn the skill, you will not get it. Okay, so it is very much important to produce a clean control, because in microbiology, none of the experiments can progress without producing or conducting an experiment in sterile manner. Even though there are significant findings in microbiology, we cannot just ignore that it all started with a candle and ethanol lab, where they have used to produce clean controls and conduct experiments. Even though clean control play a vital role, this is my recent class played where two fungi are fighting with each other in clear separate zones. And here is a famous plate, Alexander Fleming's plate, where we can clearly see the penicillium fungi producing a layer of penicillin antibiotic, which inhibit the growth of Staphylococcus aureus. This is actually a contaminated plate Fleming about to throw away, and accidentally end up discovering the penicillin. Okay, even though there are many significant discoveries, but without clean control, you cannot perform any experiment. See, even though they looks like a quadrant stick, can you see a isolated colony, micro colonies and macro colony growing together? This is not clean control for sure. This is a um, mixed culture. There are two different colonies happening here. This in turn is a clean culture. Okay, they don't have a mixed colony here. The colony volumes in the last quadrant is same, and there is no any other pigmented colony or anything. So, anytime you work on microbiology, you are supposed to have a clean control for its comparison. Even if you are working on a broth, you are supposed to have a uninoculated broth as a control, which after 7 days it should remain, transparent. Turbidity usually represents the growth of the inoculum. Okay, if you want to be satisfied that what you have inoculated is actually growing, you need to have an uninoculated control for comparison. All right. So here, uh, if you are someone who is actually spreading the plates like this in the laminar, you are bound to have contamination, especially with the baseline. Okay. So according to me, there are three ways you can prevent contamination in the lab. This is you are blessed if you got this early in your microbiology career instead of waiting 4.5 years. 3 years BSc and 1.5 years in MSc. At the last semester, if you are going and sitting in lab and are trying to do an experiment, you are not going to do anything. Okay. So unless until you have the skill of producing clean control, it is not feasible to do any experiments in microbiology. Before getting into the bottleneck points that needs to be focused for getting a clean control, okay, I will talk about these three things. Okay, so every time you do an autoclave, make sure you are changing a water. You are using a fresh tap water for autoclaving or RO water for autoclaving. Prepare the laminar flow. Ethanol wipe it latitudinal and longitudinal ways. Okay, and have an UV lamp burning for at least 15 to 20 minutes before you sit in the laminar flow. Then the third is the most important thing. It's all entirely in your hands here. how are you handling the plates? Okay, I would say you should handle the plate in your left hand, and you should have your relatively heavier object in the right hand for pouring media or any other things. Okay, I see many students keeping the plates in the floor and pouring the media. This is simply not going to work. All right. So pouring media should be around 55 degree. I have seen many people pouring with a molten agar at around 80 degrees, 70 degrees. This is not also not going to work. I will tell you why. Okay, and ability to open and close a plate, okay, and flask around the aura of the Bunsen flame. It is very much essential. That's why you should handle the plates while pouring, not keep the plates at the floor. And finally, about the sealants, I will be talking to continue the sterility inside the plates. So you guys know the role of UV lamp, right? So UV lamp. So here is an ideal experiment conducted in our lab recently. There is a mixed culture of fungi here, but we use the broth inoculum to assess the effect of UV on these two species. Okay, this is an exposed plate to UV. This plate is exposed for 5 minutes, 10 minutes, and 15 minutes. We can clearly see all four plates are equally inoculated with fungi, but when the time of UV exposure increases, number of colonies in the plate decreases. Okay, this is just to show that how UV works. All right, now let's see how laminar works. Okay, so okay, before we see how to handle the plates, let's see how to handle the laminar. All right. So my laminar flow, we have biosafety 1 or biosafety 2 cabinets. Okay. So basically, this is a clean room where the air is sterile. Okay. You have a shutter to open the laminar. It is very much important to remember laminar should always be kept in positive pressure when opening the lid. Okay. Opening or closing the lid should be in positive pressure. So laminar is connected to a power circuit. Maybe we will connect them to a fuel source or LPG gas through which Bunsen burner is burnt inside the laminar. We have a microcontrollers for UV light, white light, and airflow. Here we have HEPA filter, high efficient particulate air filter, HEPA, which is usually less than zero point 0.2 micron pore size. The average size of bacteria is 1 micrometer. So most of the dust particles and bacteria will be filtered there. Air is usually taken either from upstairs or here through this portal way, which will be sent at the bottom of the floor, that reaches the motor pump, which pumps the air into the HEPA. Okay, so through the HEPA, the air is filtered here, and the sterile air is actually reaching inside the laminar. That's how we are keeping out the contaminated air getting into the laminar flow, and the air inside this, even though they are taking out the fungal spores or anything from the plate, they are again filtered in the laminar flow, that is not leaking back to the environment. This is how we make sure that laminar works. Okay, the important thing you need to understand, especially after sterilizing the laminar, after ethanol wiping and UV sterilization, always keep the laminar in positive pressure. The air can come out of laminar. Okay, whenever you are opening a lid, the laminar should be running. Okay, always remember that it should be kept in the positive pressure. Okay, now handling the plates. Okay, how to handle the plates. This is not the way to handle. Okay, keeping the plates at the bottom, away from the Bunsen burner, and pouring the media. This is just not the way. As you can see, it is boiling media. He has a oven mitt here, and when he pours the media, everything will be settled there. Air will be condensed, and they will form a water droplet. Once the media is solidified, when you try to open it, all water will fall on the surface of the media again. There increase the susceptibility of the agar surface for contamination. Okay, this is also not the way to solidify the media. Okay, here people have poured the media, off lid open, laminar running. They think that it is solidified. No, whenever you see Bacillus megaterium or Bacillus subtilis swarming on the agar surface, it is because of these two reasons. Okay, so then how to handle it? This is the right way to handle it. Okay, either you're pouring the empty plate or you're handling the media, just pick the bacteria. Always keep the plate in inverted manner. Use your two fingers, okay, to support the bottom plate. These two fingers should support the bottom plate. These two fingers are used to open the upper cap, upper lid of the petri plate. This small finger can be used to unplug the cotton plugs or any other material that the right hand brings. Always handle the plate in the left hand. This is the essential qualification you need to have before you sit in the laminar. If you don't have that, better you practice with the water in the open table, then you reach the laminar flow. Okay. And one more important thing about autoclaving is autoclaving starts when first whistle comes. Autoclaving do not starts when you load the cooker on the stove, okay, on the LPG gas stove. Autoclaving starts when first whistle, that is 121 degrees Celsius, has been reached uh, inside the cooker during moisture sterilization, and the pressure is more than LBS pressure. That's when the weight is lifted and whistle comes. Okay, doesn't matter from the first whistle till 15th minute how many whistles comes, doesn't matter. Better you have enough water inside the cooker and let it go for 15 minutes. After 15 minutes, 4 whistles or 40 whistles, doesn't matter. After 15 minutes, get the cooker down and let them depressurize naturally. Do not remove the weight for your hurriness. Okay, let them depressurize naturally. Then you can move the materials into the laminar flow. Okay, imagine we have done preparing the laminar flow. Now, let's see. I'm using Professor Farakna's presentation to explain how the laminar flow works. Okay, so we have cleaned the laminar flow latitudinal, longitudinal ways, and we have also done 15 minutes of UV sterilization. Then I have moved the autoclaved media into the laminar. Okay, so it is always recommended to use a blue cap bottle instead of a cotton plug or conical flask for preparing media. Okay, this is much more easy and it is much more eco-friendly too. You don't need to waste the cotton. If you have a blue cap bottle, you can simply use them like this. Okay, so see, imagine this laminar flow is actually sterile. The flows have been cleaned. 15 minutes UV has been done. Even though it is so, before you start the experiment, better clean it again. After turning off the UV, turning on the white light, turning on the laminar flow, making the interior of the laminar flow into positive pressure by using the HEPA filter. The air here is actually coming through the HEPA filter. It's all sterile air. When you kept the laminar in positive pressure, now you open the lid and insert the material and start your work. Before starting the work, better again clean the surface of the laminar using 70% ethanol. It is best if you have a spray can for preparing the laminar flow. Even if you don't have a spray can, it's also just fine if you soak the cotton into the 70% ethanol and clean each and every surface. I would say instead of just cleaning like this, you can clean longitudinal ways first, okay, to the entire flow, then the latitudinal way, okay, and you can clean it. It's always best if you use a blue flame Bunsen burner. If not, ethanol lamp is also fine. But always remember that blue flame will instantly heat the nichrome loop when compared to the yellow flame. Okay. See, he has moved the material from autoclave to laminar. He want to make sure that nothing is being carried in the paper from the environment. When the paper is wet, they can absorb many things like fungal spores in the air and many things. So he just to make sure that nothing is being contaminating, he is spraying the paper and he is folding the paper and throwing it away. Okay, ethanol sterilized paper. Now he is taking the plates. He is preparing the plates to pour the media. Before that, since he has taken the paper in his hand, he is also cleaning his hand using the ethanol. Okay, this is very much important to avoid baseless contamination. Remember, whenever your plate is getting baseless contamination, usually it is from human source. Whenever it is getting fungal contamination, maybe from the lamina. Okay, this 20 minutes airflow should be enough to kill all fungi. But if your HEPA is clogged with fungal spores, usually you will get it. Okay, but even though HEPA is clogged with fungal spores, how to have a sterile plate? Always remember to handle the plates and flask around the aura of the Bunsen flame. Around the aura of the Bunsen flame is considered as a sterile environment. Okay, so anytime you open a flask, bottle, anything, opening, closing should happen around the aura. See, this is another advantage of having a blue cup bottle. When before opening, you can also stay heat the mouth, sterilize the mouth because now the mouth region is going to be exposed to the air. So before opening it, also the advantage of handling the hand wearable temperature around 55 degrees. This you can simply handle them like this. Okay, if you cannot hold a flask that is actually containing molten agar, means you should not pour it. Okay, if you have a difficulty in moving the material immediately to the laminar flow after autoclaving, if the laminar is not available, better put the media in 55-degree water bath until the laminar is available. Okay, 55 degrees is the right temperature to pour the media, which will not, see, he is also sterilizing the mouth of the media, petri plate before opening it. I would say never hold the media like this. You just hold it near to the Bunsen burner once the mouth is open. Okay, but anyway, sterile air is coming from Nepal. He is confidently holding it. He is using these two fingers to open the upper lid of the upper cap of the petri plate, and he is going to pour the media into the lower lid. Okay, while pouring, after pouring, you can also do a zigzag, infinite motion to for even spreading of the media at the bottom of the petri plate. Okay, so always remember opening and closing the media or petri plate should happen near to the flame. I would say even you can keep it parallel to this flame, not above the flame. Okay, so pour the media. Did you notice here the upper lid remains transparent? They are not clogging that much because of high temperature because it is hand variable temperature. See, you can also hold like this. So it is 55 degrees Celsius. You That's the reason actually people pouring at molten lava like molten lava into the plate. Now if you're pouring it at 80 degrees Celsius, you cannot hold it here. You have to hold it by mouth. The chances of contamination is high. Once you are pouring, these two fingers will be burnt. That's why people are keeping on the floor of the laminar flow and getting all base like contamination. Okay, so once you learn how to pour it around 55 degrees Celsius, you can naturally pour it and make the plates here. Okay, um, the agar will solidify when the temperature falls below 45 degrees Celsius. They will start solidifying. So I think this 10 degrees is a buffer temperature that gives you enough time to pour at least 5 plates instantly. If you have preparing say 1 liter of media, multiple plates, I would recommend prepare it in 4 volumes of 250 ml. Keep remaining 3 bottles in 55-degree water bath when you are dealing with the first bottle. Okay, that's how the plate should be made. I will guarantee after 4 or 5 minutes when the temperature drops below 45, these plates will solidify without containing any moisture in the upper cap. Okay, so this is how a media should be opened and poured. If you have a blue cap bottle, better use it. It is an autoclavable bottle with autoclavable blue cap. All right, it can, for storage also reheating. Many things are easily done in the blue cap bottles. Okay, so that's how plate should be made, and that's how you will avoid contamination. Now, if you want to inoculate some samples, you can open one plate, leave it at the corner of the laminar flow to check whether the laminar is being contaminated. Okay, you can plate remaining 4 plates, and when you're done with the plating, close the control and incubate the control along with the other plates. This is called clean control. Even after 7 days, it should remain clean. No bacteria should grow. If you have prepared the laminar from the autoclaving, change of water, how the autoclaving is done and how you have prepared the laminar, how you have handled the plates around the Bunsen burner, around the flame of the burner. Everything decides whether you will get a clean control or not. Okay. Finally, there is a sealant. Okay, this is the small work I have done. I have witnessed this results. Does sealing Petri plate influence bacterial growth? You can check this through Google. You can get this article from ResearchGate, where I have plated this eight different bacteria in the nutrient agar. These plates are sealed one. These are control plates. Other eight are plated here, and these are control plates. This I have not used any sealant here. I have seen there is no difference in bacterial growth. That's the conclusion of this work. Okay, so what's a need to use a sealant? In my life science lab, I have my Drosophila lab happening near to the incubator, which will attract many flies into the place. So if you have a sealant, it will prevent you from the flies. And also, especially in academic setup, when you have hundreds of plates piled up inside the incubator, simple opening the door, with a fast manner, if you just pull the door out, you may end up in sliding all the plates and breaking the glass petri plates. Okay, so once you have a sealant around the plate, it causes a friction. Simple gliding, accidental breakage is very much reduced if you have a sealant around the plates. So I would always recommend to have a sealant. So don't try to produce a clean control during your last final semester. This is something you should earn it during your first BSc microbiology. Okay, you should have done this. If not, it's never too late. Practice in open table as much as possible before you sit in the laminar flow, because any work requires pure culturing. Without clean control plates, you cannot end up doing pure culture, which is a step one for any microbiology work. Thanks for watching.